So I'm going to give you some impressions of what we have done in the last few years, trying to, in the context of anti-inflammatory drugs. So this is the region where I am. So in the Midwest, in the southeast Brazil. So I will actually try to discuss three things: the general concept of actually developing anti-inflammatory drugs, what people refer to as also as host-directed therapies, the new concept that's emerging in the field of inflammation, which is the resolution of inflammation, what it is and how we can use it to treat inflammation and eventually infection. And then how do we apply resolution pharmacology for the treatment of infection? So I'm actually, just so you know, in Belo Horizonte. It's 1 hour flight to the major cities of Brazil: Rio, São Paulo, and Brasilia. So it's in the southern easternmost dense part of the country. So Belo is about a 5 million region metropolis. Okay, so the work in my lab, we are interested in understanding the inflammatory signals and how these are controlled, the inflammatory signals that drive leukocyte influx, and how actually leukocytes change functional response during infections, disease, and also chronic inflammation, obviously focusing here in infection. And the leukocyte will change functional response in infection by facilitating or decreasing establishment of infection, decreasing replication, and also changing the onset of disease, even if infection is established. So just to describe briefly, most of you don't know the work we do. And obviously, as you are a large antiviral center, I'll just present some of the things we do. Our major strength has to do with doing animal models of infection. So we do, these are established models in the lab: dengue, influenza, beta coronavirus, yellow fever, Mauro, congenital Zika, chikungunya. So these are actually up and running in my lab. And I suppose the strength that we have is that in addition to actually looking at viral replication, so this is chikungunya given into the paw of mice. Obviously, it stays to about 7 days, so we can measure viral replication and how that actually is seen in terms of the inflammatory response, such as swelling, influx of neutrophils and macrophages, and then you can do histology. But very importantly, how that actually changes function. So Mauro is a disease that causes arthritis, so we can actually do experimental tests that we measure pain in animals, and then we can actually see some dysfunction associated with infection, which is arthritis and arthritic pain up to 21 days of infection. So any treatment, any vaccine or drug would be seen not only in terms of its effects in the viral load, but also in the leukocyte influx and the eventual impact on that on a functional response. During that, we have collaborated with different groups throughout the world, trying to develop mostly anti-dengue or anti-flavivirus compounds. This is not our major strength. It's really to interact with these groups and actually see the real impact of these strategies in animal models. The other side, NMMD, and we do quite a lot of clinical studies, a lot investigator-driven, and so I mentioned a few of these. So we do mostly clinical work on these infections: dengue, chikungunya, influenza, and COVID. A lot to do with vaccine trials, but also antiviral drugs or anti-inflammatory drugs, as a matter of fact. And we also have a very large NIH-funded study looking at Wolbachia and the impact of Wolbachia on the amount of dengue in Belo. So pretty much going back to our major interest. So and I like very much this figure. So we are actually, surrounded by a huge amount of microbes. It's estimated about 1 trillion species on Earth, most of which we don't simply interact, right? So we are amidst them, but we have no molecular counterparts, so we're not infected. And presumably the most common interaction we have with the microbial world has to do with the microbes that actually live in our gut. There are about 400, 500 species of bacteria plus viruses, archaea, and fungi that live in our gut, and they're actually very important. So indeed, our life is all about interactions, not only with our microbes, but also with that outside world. And that interaction is very important because, the metabolic world, it actually complements our genes, it complements our metabolic capacity. And from our point of view, it actually is necessary to this maturation of our immune system. And so those microbes in our world actually help us to adapt to the environment. So, and the way we see it is by actually having those microbes, it actually helps us to adapt to the world. It helps us mature our immune system. And depending where we live, who we interact with, what we eat, that actually changes our microbes. So in a way, that microbiota is almost the environmental sensing of the immune system. The microbes change in response to the environment, and that actually talks to our microbiota, to our immune system, and changes our ability to respond in many ways. So this is something that we've been working with for quite some time. And why do we think it's important? If we think of our interaction with microbes, both inside and outside, it's very much like our social interactions. So this is a market in Thailand, but it could be anywhere in the world where you have a lot of people. So life is indeed about interaction, and any form of life is about interaction. If you see a tree here and you look at the other picture I had, it's all about a lot of pressure on us, and we need to interact. One form of interaction, and this is the major topic of what I'm talking about, is when that interaction leads to infection and eventually to disease. So there are estimated about 1,500 pathogens that infect humans, many of which are viruses. And the major interest about viruses is that we don't have a lot of treatment to existing viruses. And if we look again in this picture, a lot of our interaction is normal like interaction, so we just simply do not interact. Then resident interaction, infection, eventually disease are really the tip of the pyramid or the iceberg. But infection does matter because that's what takes people to the hospital. But and it's interesting that we look at the immunological responses of individuals during disease, but very little to the resident microbiota or only to infection. This is what I say, it's almost like studying human social interactions by studying war only and not what happens under normal circumstances. But interactions which cause disease matters a lot. And this is why we want to develop drugs that actually will prevent disease. And so this is a very important point to start discussing. So we may interact with a microorganism. It may infect us, replicate, and eventually cause disease. So infection and disease are two different things. You may be infected and not have disease, whereas you may be infected and have disease. And what we want to prevent as doctors is actually disease, not necessarily only infection. So we have hypothesized that, well, it's an observation. During infection, there will be production of mediators of inflammation. And these mediators are necessary so that you contain replication and then you mount adaptive immune responses that will eventually contain the infection that is occurring. Now, disease will occur if these mediators are produced in an inadequate way, meaning insufficient, excessive, misplaced, and altered. Different diseases will have different occurrences, like HIV. When it's advanced, you may have disease to a secondary microorganism because there is insufficient immune response. Sepsis is a well description, and COVID-19 as well, of excessive inflammatory response and perhaps misplaced because it's very systemic. Now, if disease results from inadequate production of mediators, it is possible that we identify mediators that are produced during infection that may be relevant for disease and not for the protective immune response. And if we can identify those, we can actually develop anti-inflammatory drugs to treat infection. So This is the unifying concept of the studies in my lab, and that we have been trying to do that for many years now. So this is a review from 2010 that we have been trying to pinpoint and find anti-inflammatory drugs that could eventually be useful for infectious disease, and especially in the context of arboviral infection, because of the relevance of these for Brazil. Obviously, and this is one example, for example, is an example that's found. So in mice infected with certain dengue strains, there will be thrombocytopenia, hemoconcentration that is decreased, or in the presence or in the absence of the PAF receptor, or treating with PAF receptor antagonists. And indeed, if you treat with a PAF receptor antagonist, even very late into the infection, you see some degree of protection from death in these animals. So in the context of experimental dengue, blocking PAF receptors will prevent disease without actually interfering with the ability of the host to deal with dengue infection. And I think now there is good evidence for that concept, that blocking aspects of inflammation will be beneficial as an adjunct treatment for many infectious disease. And I think COVID-19 brought very, very important clinical evidence, even with steroids being very life-saving in many patients with severe COVID infection. Okay, so that is the concept we're working with. It is indeed now accepted that the anti-inflammatory drugs may be useful as joint treatment for infectious disease. This is what we usually refer to as sterile. Now, I will present to you a concept that's new to many people outside the area, which is the concept of resolution of inflammation. So it's become clear in the last few years that we have a very active process to resolve inflammation. That active process involves, killing of the cells of leukocytes that are in tissue and activated by inducing apoptosis, and then eventually eating those dead cells, a process referred to as efferocytosis. As you're doing that, you actually produce molecules which will dampen down the arrival of new cells, reprogram macrophages, and eventually you will enter a new homeostatic state, which is close to your naive state previous to that inflammatory stimulation. Now, the realization, which is very strong in the last few years, is that resolution is not a passive process. It doesn't occur because the stimuli have gone away. It is very active. You need the active production of chemical signals and interaction of cells, which will drive that resolution. So we refer to these molecules in general as mediators of resolution of inflammation. And my group has been very interested in this. A definition of that would be, it's an active process brought about by biosynthesis and release of active mediators, which act on key events of inflammation to promote the return to homeostasis. So the concept of resolution is very interesting from the point of view of treatment, because it's not about prevention of, it's about treatment and return to homeostasis. And we have been very active trying to find these molecules. And our studies in germ-free mice actually suggest that nature has chosen resolution. I don't have time to show you the evidence, but nature has chosen pro-resolving molecule as a major means of germ-free mice to be non-inflammatory under the germ-free condition. So if I stimulate germ-free mice, they produce large amounts of pro-resolving molecules, and they are anti-inflammatory from that point of view. So we have been describing many of these mediators of the resolution of inflammation. Obviously, the whole field started with Charles Serlin in Harvard, actually describing the lipid mediators. But this is to make the point that it's much more than that. The lipid mediators are strong resolvers, but there are proteins, peptides, gases, short-chain fatty acids. There are a range of molecules now that have been described. They are produced late in the late phase of the inflammatory response that will drive the inflammatory process to a close, that will make inflammation faster. So the concept in the context of chronic inflammation is that, for whatever reason, there's failed resolution, and that failed resolution will drive chronic inflammatory disease. In contrast to the usual acute inflammatory response, where you have the rival cells, production of mediators of resolution, and then return to homeostasis. Now, the new concept that arrives from that, you may actually come up with the molecules I've told you about, is that you may give them back to individuals, and by giving them back, you may actually restore the capacity to induce resolution of inflammation, what we refer to as resolution pharmacology. Okay, so the new concept of resolution of inflammation and resolution pharmacology begs the question whether we can use that concept to treat inflammation or infection in such way that can resolution pharmacology help protective immunity and dampen down unwanted inflammatory responses. Okay, I'll show you a few examples of resolving molecules and their receptor in the context of infection. The first one is annexin A1, which was discovered as a steroid-induced molecule. Annexin A1 will bind to a receptor, 7-transmembrane receptor. In fact, all of these receptors are 7-TM receptors, FPR2, MAS, or MCR1. Annexin 1 will bind to FPR2 to mediate its effects. So if we infect mice with a clinical dengue 2 strain, what you will see is that very rapidly you see a decrease in circulating levels of annexin A1. It is metabolized, it's cleaved. So you have that decrease. Although the receptor, the agonist is gone, the receptor is there. Actually, in such way that in mice, you actually have upregulation of the receptor. So there is a very interesting therapeutic opportunity where you have lost the ligand, and you still keep the receptor there in many cell types, including especially macrophages, not shown here. Now, if you focus on letter B, right? So because there's a lot of data, so I don't want to confuse you. Now, if you infect these mice with this clinical strain of dengue, they will have hemoconcentration because there is increased vascular permeability changes. In the absence of annexin A1, it's like removing the brake, right? You have a more intense and a more severe and more prolonged inflammatory response to that strain. You know, these are immunocompetent mice, so the disease is very short because there is no good replication of dengue in these mice. There is more of a response to the virus that you inject. But in any event, we have a more intense and more prolonged inflammatory response when you remove annexin 1, you remove a brake, or when you remove the receptor, FPR2. Again, if you remove the FPR2 receptor, there is more intense and more prolonged inflammatory response to dengue. There is more response, there's more disease, cytokines, you name it. Okay. In contrast, if I give annexin A1 back to these animals, if I give annexin A1 in excess to these animals, what we observe is that there is much less disease. There is less, there's less hemoconcentration, thrombocytopenia is reversed, and there is less vascular permeability changes and cytokine changes. So giving the agonist back is protective only if the receptor is present, because if you do experiments in the absence of the receptor, protection is lost. So you have an NEXI1 analog will protect via binding to FPR2, right? The disadvantage of this model, immunocompetent mice, you cannot look at viral load. Very short, you cannot look at the therapeutic schedule. So we shift to A1 to 9 mice. Again, here you see protection. You give the mimetic, you see less thrombocytopenia, less body weight loss without any major effect on the viral load. So we are not changing the capacity of the host to deal with infection. We're changing the inflammatory response of the host, and that is beneficial. That will reverse in cytokine changes, less cytokine changes not seen here. And very interestingly, you can actually do experiments with dengue 1, 3, or 4, and you will also see a protective effect. So it is not virus specific, it's host focused, so it will prevent the response of the host to any infection. Mechanistically, what the annexin-A1 does, it seems to be acting on mast cells. It will prevent the degranulation of mast cells when you present it to the virus. So there will be less release of granules or less binding of the secretory granules to the membrane if you do that by electron microscopy. So you decrease the granulation, but not cytokine production by mast cells. And also there is an effect because in general, macrophage or monocytic cells are very targeted by annexin-A1 because the receptor is there. In these cells, you actually prevent their activation state. You prevent cytokine release from these cells, but not the replication of the virus within these cells. So it seems that annexin-A1 will act on crucial cells associated with the inflammatory response in these animals, but not the ability of the host to deal with infection. And interestingly, I showed you that in mice, there is a decrease in annexin-A1. So we followed dengue patients. And again, we see that very acute, this is day 3 to 5 during, into the infection. You see a decrease of annexin-A1 in dengue patients. And interestingly, if you look at dengue patients who are more or less severe, the more severe you are, the lower your annexin-A1. So it seems that in dengue patients, you mimic the situation in mice. So what I've shown you, if you remove annexin-A1 or you remove the receptor, disease is more severe. If you add annexin-A1 in greater amount of pharmacological doses, you protect mice from disease. And it seems you have a similar situation in humans, suggesting that humans may be amenable to treat with this kind of peptide or molecules which mimic that peptide. Okay, so another example, which I'll show very briefly, is angiotensin 1, which is also, it's a short peptide derived from angiotensin 1 to 2. It's actually cleaved from angiotensin 2 to angiotensin 1 to 7 by the angiotensin converting enzyme 2. So that's what it cleaves to this peptide, which in general has beneficial effects on the cardiovascular system. It has antihypertensive, prodiuretic, and anti-inflammatory effects. And it does so by acting on these 7 transmembrane called MAS receptor. So in the context of influenza, if you give this peptide per mouth, this is a formulation with cyclodextrin, you will actually prevent disease. As you see here, animals die less. They die less because the lung is less inflamed. The lung is less inflamed because that molecule will kill neutrophils by inducing their apoptosis, and eventually, not shown here, their efferocytosis by macrophages. And when macrophages eat up dead cells, they don't get activated. They don't induce further damage. And the overall inhibition of neutrophil influx will be associated with a lesser inflammatory score in the lungs, which will actually cause less death. And interestingly, if we use a small inoculum of influenza, right, that does not kill on its own, but actually facilitates S. pneumonia infection later. So if influenza infection followed by pneumococcal infection kills all of the animals. Whereas if you do the same pneumococcal infection naive animal, nothing happens. So there is facilitation of infection, pretty much as we saw in the influenza pandemic. You can actually decrease that by simply give angiotensin 1 to 7 during influenza infection. So you actually changing the whole course of influenza infection by giving this pro-resolving molecule, because it makes your not shown here, the macrophages respond better when issuing pneumococcal infection. So again, another example in which you can give a pro-resolving molecule to a viral infection, and that will change the course of infection, right? So I hope I've shown you at least 2 examples. There are others I've not shown you. We have data for chikungunya with annexin-A1. we have data for angiotensin 127 in models of beta coronavirus infection as well. So I think there is good now proof of concept suggesting that resolution pharmacology may be useful as an anti-strategy. Also in sepsis, actually, I'm not showing you any of that, but in the context of bacterial infection, these molecules even facilitate the ability of the macrophage to eat and kill bacteria, which is actually very interesting because bacterial infection very frequently follow viral infection. So the final example I want to show you has to do with this molecule called AP1189, which acts on melanocortin receptors 1 and 3. AP1189 is a drug currently in phase 2-3 trials for the treatment of arthritis. It is a biased agonist, so it will act on those receptors. It will not change the color of skin because it doesn't activate the cyclic AMP, but it will have very strong anti-inflammatory effects. Now, so we've done an investigator-driven study in COVID patients, phase 2, in which we asked the question, if I give AP1189, will I decrease oxygen usage by these patients? So the primary endpoint was actually asking individuals, how long does it take for me to remove oxygen from you? So I'm going to give oxygen. Everybody is arriving with oxygen, with the need of oxygen. If I remove, how long does it take for me to go back to breathing on room air? So we actually triaged about 400 patients in this particular hospital in Belo, and eventually treated 60 with placebo and 44 with, 42 with AP1189. So there was a phase 2 in which we gave, we looked at the safety of giving this drug to patients, and then they were randomized 2-to-1 to AP1189 or placebo. So the primary endpoint, what we named days for respiratory recovery, as defined by how many days does it take for me to remove oxygen to keep a saturation of oxygen above 94. And you can see that there is a major decrease in the number of days actually individuals stay using. So there was on a median of 6 days in placebo, 4 days in the AP1189, because individuals used less AP1189. They recovered faster. They went home slightly faster, about 20%. And there was a trend throughout of better outcome in terms of ICU admission, mechanical ventilation usage, days in using the ventilator, right? So you can actually put that in graph. So basically, individuals taking AP1189 are better off because they tend to go home earlier than individuals being treated with placebo. And overall, those effects of AP1189 are associated with inhibition of cytokine production by macrophages. I didn't have the time to show you that. But I think this clinical study, although it's short, is the first actual demonstration that resolution pharmacology may be a useful anti-disease strategy in humans. So I will leave you with these 3 take-home messages. Indeed, we can use anti-inflammatory drugs to treat infectious diseases. And we can actually use this new concept, which is called resolution pharmacology, and apply it for the treatment of infection in humans. There are many people I have to thank. I don't have time to name, but these are the people who've done mostly of the in vivo and in vitro work and the clinical studies in Belo. And I thank you very much for your attention. Thank you.